The series of Ocean Acidification, Harmful Algal Bloom webinar series. My name is Beth Turner. I work for the NOAA National Centers for Coastal Ocean Science. I'll be moderating the webinar today. Jennifer Mintz from the NOAA Ocean Acidification Program is our producer. She's going to be working to keep Adobe Connect going. If you have problems with Adobe Connect, you can enter them into the chat box in the upper right corner of your screen, and we'll try to address them. And Jen will be monitoring that as we go along. Thank you for joining us for Alaska Ocean Acidification and HABs, Networking and Coastal Variability. This is number 8 and our final webinar in the series of webinars that's preparing us for our virtual HAB OA workshop on August 11 to 13. At that workshop, we're going to be discussing HAB OA interactions, the major questions surrounding them, and the research priorities that can help address gaps in our understanding and management of them. The workshop will help to identify priorities and research products that could be incorporated into a future request for proposals on the intersection of HABs and OA. If you're not yet signed up for that workshop but you'd like to participate, please contact Maya Sharp. Her email is going to magically appear in the chat box when Jen puts it in there. And I'm going to let Maya introduce herself and share a little bit about her summer project with us.
Thank you, Beth, and hello, everybody. My name is Maya Sharp. I'm a NOAA Educational Partnership Program undergraduate scholar, and I'll be completing a project based on webinar's content, questions, and discussions. The chat box will be monitored and recorded during the webinar, and there will also be a questionnaire that we will use to inform our workshop and future directions of research funding. Those accessing the webinar online will have the opportunity to comment and ask questions through the Google form, and the link will be in the chat box for discussion at the workshop. I will compile these and synthesize them for our workshop. Thank you.
Thank you, Maya. The questionnaire that Maya mentioned is in the chat box. Jen here, I just wanted to take a few moments to help us all get oriented in Adobe Connect. So audio is over the computer. You may listen through your computer speakers or a headset. If you're having any trouble hearing, you can either try turning up your volume on your computer speaker or headset, and you can also turn up the volume in Adobe Connect. As you can see here in the top menu bar, you can click the pull-down arrow to the right of the speaker icon and select Adjust Speaker Volume. If you are looking for a particular speaker or headset, go to that same icon, hit the pull-down, and select Speaker to find the headset that you are looking for. If you are still having any trouble hearing, we do ask that you log off and log back in using the Adobe Connect application rather than the browser. During our presentation today, you can view it in full screen mode by going to the button highlighted here, and that is the 4 arrows in the upper right edge of the slides. This button toggles the slide size larger or smaller. Please note that you will lose chat and captioning boxes while in full screen view, but can toggle back to the regular view at any time to ask your questions or add comments to the chat box. We do ask that you enter any questions you have into the chat box highlighted here, and we will hold all of those until the end of our presentation. As you can see, we also share relevant links in this box, including that questionnaire that Maya mentioned. And we can't encourage you enough to just take a few minutes after the presentation has ended to go ahead and fill out that questionnaire to not only support Maya, but allow us to be more informed as we move forward in OA and HAB research. At the end of today's presentation, we will open it up for questions and discussion. If you want to comment but aren't the questioner, you may go ahead and raise your hand by clicking on the icon highlighted here. We'll unmute you, and you may have that conversation with our presenter. And we will be posting a recording and a PDF of the slides on the workshop webpage, which the address of that can be found in the chat box, along with the link to these questionnaires that have accompanied each of the webinars in this series. All of the webinar recordings will be available on that website at the end of this week, and we do appreciate you not only viewing those that you have missed, but taking a few moments to respond to that questionnaire so that we can have a robust discussion at our workshop. Beth, I will hand it back to you.
Thank you. Today we're very happy to welcome Kris Holderied. She's the director of the NOAA Kasitsna Bay Laboratory. In that position, she oversees facility research and operations. She conducts research on coastal ecosystem change and supports marine science education activities. Kris's research interests are in oceanography, estuaries, and nearshore habitats, and she focuses on a better understanding of how changing ocean conditions, especially associated with climate change, harmful algal blooms, and ocean acidification, affect Alaska coastal resources and communities. Kris previously worked as a physical oceanographer with NOAA's National Centers for Coastal Ocean Science in Silver Spring, Maryland. She developed satellite-based products for benthic habitat mapping, harmful algal bloom detection, and coastal climate change impacts. Before that, she worked on environmental compliance projects for the U.S. Army Corps of Engineers in Norfolk, Virginia, and she served as an active duty oceanography officer in the U.S. Navy. She has a BS degree in oceanography from the U.S. Naval Academy, and an MS degree in physical oceanography from the Massachusetts Institute of Technology Woods Hole Oceanographic Institution Joint Program. Kris, we're really happy to have you, and please take it away.
Hey, thanks, Beth, thanks to Beth, you, and Jen and Maya for your help in organizing this. You make it quite easy to do, which I appreciate. As Beth said, I'm Kris Holderied. I'm director of the Kasitsna Bay Laboratory in South Central Alaska. And I've been with NCLAS since 2000, and I've had the good fortune to be up here in Alaska at the Kasitsna Bay Lab since 2005. And I today wanted to give you just a flavor of some of the things that we're dealing with for OA and HABs in Alaska, kind of focusing on the motivation for what were the issues that we're dealing with, some of the ways that we're coming together to network and coordinate better around those issues, and give you some examples of the variability that we're seeing in our coastal waters here that, complicate all of the above. And let me make sure I do the right one. Oh, I'll pop back. And then I'm talking about work, just a slice of work that's being done in the state, and have acknowledged the support from the Alaska Ocean Acidification Network and the Alaska Harmful Algal Bloom Network, the large group of researchers and stakeholders working in those networks and coordinating through that. It's really been moving forward in the past 3 to 4 years on the coordination has, I think, made a big difference in how we're approaching these issues now in the state. But I'd like to say, while these issues are core missionary areas for us at the lab and within NOAA, it's also personal, right? I live in a small Alaska coastal community. I like to fish and clam and eat, the best oysters on the planet here. And I've also seen the changes in the time that I've been here in the declines of shellfish, in having more paralytic shellfish poisoning events pop up, and seeing changes in the fisheries. So it's kind of both a personal and professional interest for me. And the marine resources really are what our coastal communities depend on. So whether that is fisheries, whether that is subsistence harvest for food, whether that is mariculture industries like the oyster farmers, or ecotourism, there really is a heavy dependence on the marine resources here in the statewide. And those resources are supported by just really rich coastal ecosystems, whether it's the near-shore food webs from kelp to shellfish to otters and shorebirds, or the pelagic food webs from the plankton to the forage fish to predator fish and whales and seabirds. So the motivation that we have is really looking at how these ecosystems are responding to environmental changes. And we've been seeing a lot of environmental changes, particularly in the past, well, really since the 1990s. But particularly some of the examples recently, the blob, the Pacific marine heat wave that affected all of the North Pacific and the West Coast and Alaska, really was dramatic here in terms of increased paralytic shellfish poisoning events, die-offs of marine birds and whales, and complete crash to some key fisheries. So recent events have been pretty dramatic, and then we also have longer-term changes that we've seen in declines of shellfish, crab, shrimp, clams since the 1990s. So long-term changes as well. And so our motivation is we're looking at HABs and ocean acidification changes, but it's in the context of a lot of these other changes, particularly with respect to the climate changes, both on long-term and kind of decadal scales. And then we're also, there's a lot of oil and gas industry and other development in these same coastal areas. So really the communities are dealing with all of this, and we're kind of looking at them as intersecting issues. And then the next complication, of course, is that we have a very large coastline with remote and widely separated locations for the communities. We have 5 different large marine ecosystem regions around the state, from the Gulf of Alaska into the Aleutians, Bering Sea, Northern Bering Sea and Chukchi, and the Beaufort Sea. And the coastal areas in these ecosystem regions are very different from each other. So we're dealing with that spatial complexity as well. And for scale, Cook Inlet here, which is circled on in south central Alaska, is about the length of Chesapeake Bay. So just for that, I'm not going to talk about things across the entire state. It'd be a much longer talk. But we'll focus on a couple of things in kind of our backyard in Cook Inlet and Ketchimak Bay in south central, as well as some very different areas up in the coastal Beaufort Sea and the Beaufort Lagoons. And they give you a sense of what things look like in our backyard. So this is some of the imagery of habitats in Ketchimak Bay, and very rich, diverse habitats of kelp and seagrass and watersheds that are fjords, as well as braided glacial rivers and watersheds. So really a lot of complexity in small areas, which feeds into what we have to understand in order to be able to deal with both harmful algal blooms, how they're changing, how they're affected by the environmental conditions, as well as ocean acidification conditions, and then the intersection between all of the above. So, and then I'll show you some examples of data from Ketchimak Bay, but for contrast, in terms of what something looks like, this is now an aerial shot of the Beaufort Lagoon ecosystems up on the north slope. And there's a large study going on there right now, funded by the National Science Foundation, called the Beaufort Lagoons Long-Term Ecosystem Research Program. And so these are very different systems. They're shallow. They're very, very productive, but go hypersaline and are covered by ice in the wintertime. So just very just to give you a sense of a couple of the different systems that we're looking at within the state. And I'll be showing some ocean acidification data from the Beaufort Lagoons, courtesy of Dr. Amanda Kelly from UAS later. So stepping back to Ketchikan Bay, it's, I mentioned before, it's kind of a microcosm of the different habitats and watersheds and coastal influences that we care about for things that affect both harmful algal blooms and ocean acidification. So whether that's temperature, salinity, turbidity, nutrient changes, all of the above, we have that variability in this relatively small area. We also have the advantage that there's several research and management agencies that are co-located here. So it's our Kasitsna Bay Lab, the Ketchikan Bay National Estuary Research Reserve, University of Alaska Anchorage and Fairbanks both work here, as well as offices of the Alaska Department of Fish and Game. And then we're also connected through the networks and other organizations to the statewide management agencies and other research organizations. So, and the places I'll show you some data from here are the yellow stars are where we have water quality monitoring stations run by the Ketchikan Bay Research Reserve that have been in place from the early 2000s to present at the Seldovia and Homer harbors, as well as in Bear Cove at the head of the bay. And we have a lot of attention paid with the heat wave and the long-term climate warming and decadal warming patterns to temperatures. But the Alaska coast and ecosystems are equally influenced by freshwater changes. And all of the issues that we were talking about with respect to food web changes in fisheries, shellfish, the HABs and the OA, they are also influenced by these freshwater changes. And there's the Alaska unique pieces of that, where we're dealing with sea ice and melting glaciers and snowpack seasonality. And those changes are just like temperature coming in at both a local scale, so at the scale of the bay, but we're also influenced by changes on a coastwide scale. For instance, the freshening of the Alaska coastal current, which runs along the Gulf of Alaska, will influence our local conditions within Ketchikan Bay. So we're kind of always looking at these different scales, both in time and space, and need to characterize them to understand really what's going to be driving both changes in HABs and ocean acidification. And so one of the way I'm going to talk about how significant the change was, a recent change we had was in precipitation, was this weird word that showed up last summer that we really don't see in our part, which was drought, right? So we were concerned, and locally we were concerned about we had a huge wildfire, and we had drinking water shortages in the local Alaska Native villages in particular. But those same changes over 3 months were enough to change the salinity conditions within the bay, at least for those areas that weren't influenced by increased glacial melt from all that hot, sunny weather. So that's just a little bit of the sense of kind of the intersection of some of these forces here. So kind of shifting from that background and motivation and kind of the environment that we're looking at to how we're trying to come together within the state, particularly since the past 3 or 4 years in developing some very robust networks of researchers, stakeholders, managers, industry folks, tribal organizations. It's really a diverse mix of coastal stakeholders within the Alaska Harmful Algal Bloom Network. And the objectives here, as shown on the right there, it's really about helping with the needs assessment, figuring out, hey, what do we know, what do we not know, what's the emerging threat, what's happening in the Arctic right now, how can we use what we know about longer monitoring efforts that have been going on in South Central and in Southeast Alaska to inform how we could set things up in Arctic communities that haven't had to deal with these threats before. So looking to improve event response coordination, looking to develop risk assessment and ideally in the future event forecasting capabilities, and do a lot of informing of people at all different levels. So whether it's decision support products for management or public outreach and education, or very specific public advisory information for subsistence harvest, which is that's a big topic right now because we have some very, very high levels of paralytic shellfish poisoning in some parts of the state right now. So it's helping on all of those issues is really what the network is doing. And with the goals of reducing human health risk and helping to keep those seafood supply safe, whether that's subsistence or mariculture. And so if one of the things that the network has been involved in is really in trying to improve monitoring of HABs and also trying to understand the drivers of harmful algal blooms. And a couple of recent papers that have come out, one is Van der See et al. That was based on work in Ketchikan Bay, and then Tobin et al. That was based in Southeast. And so these are really taking some of the monitoring work that's been going on for longer periods of time, and really for us taking advantage in the sense of the marine heat wave and what happened there. So the graph there is of Alexandrium cell abundance, and the dashed red line is the abundance at which you expect, and we did see toxicity from the saxitoxins in the Alexandrium cells to show up in shellfish. So that period on 2014 to 2016 was when we had much higher than normal temperatures, and we saw the pop-up in the Alexandrium that we hadn't seen in a decade. So we saw very consistent temperature relationships in our part of Alaska, in South Central. We didn't see the connection with salinity. The work done by Tobin et al. Actually showed very much better connections with both temperature and salinity. So we're looking at regional differences there and then trying to understand that by sharing information across the network. And then, it's like last year in 2018 into most of late 2018 into 2019, we ended up, hey, it was warm again. We're warm. We're like, okay, we're moving into this, another heat wave. We're ready for it. We're ready for PSP and all of that, and this is what happened in January. We went anomalously cold, super cold. So the winter, instead of being a warm winter, turned rapidly cold. So just the sense of, the range of conditions that we're dealing with and trying to understand how this kind of variability affects both particularly for harmful algal blooms, but also the ocean acidification conditions. So, and then stepping out from Ketchikan Bay back out to the larger state, just to get a sense of, well, what happened in the, during the heat wave years and how widespread that was across. So this graph shows it's from it's going back 117 years of sea surface temperature data and ranking those years. So if the color is red, it means that these years, the 2016 was the warmest year in the entire record for everywhere that it's red. So that's the graph on the left, and then the plot on the right is an average temperature again, over that same period, so from the 1900s through 2016, again showing the anomalous, how anomalous that marine heat wave was in 2014, 2016. But following that, 2017, Gulf of Alaska kind of went back to, you know, we're only in the 19 to 30 sort of hottest years in that record, but still in the Bering Sea it was quite warm, and what happened subsequently is that we ended up not having winter sea ice in the Bering Sea, which was a really dramatic change. So again, just a sense of kind of the variability at the larger scales, which intersects with the variability happening in the bays and estuaries. And so stepping back to some of that variability within Kachemak Bay, so looking at kind of, you saw where we sat across all the Alaska waters during the heat wave. This is now from the water quality stations at the Homer Spit, at the Homer Harbor. So it's a time series going back to 2002 through last year. And red, the top plot is temperature anomalies, water temperature anomalies, and the bottom plot is air temperature anomalies. These are monthly anomalies against the entire mean. And so the red bars are warmer than average, blue bars are colder than average, and you can see the 2014, 2016 warm blob area, and then popped up again in 2019. Difference between those being the warmest times during the heat wave were actually in the winter months, and the warmest times we had last year were in the summer months. So we're still looking at how that is influencing HABs in particular. And then just you get the sense of the decadal changes here, as well as the close connection with the meteorological changes. So the drivers coming from large changes in the Pacific. And then also showing a similar plot on salinity, and again the 2014 to 2016 marine heat wave. It was warmer. It was also fresher, which makes sense because of the increased, particularly increased glacial melt during that period. And the entire, this is for, again, in Kachemak Bay, but the entire Alaska coastal current was freshened during that period. So then how does this intersect with, kind of where we are on the emerging HAB issues? And I mentioned Arctic changes before, and one of the things that we saw in 2018 and 2019 with Don Anderson and the others, some of the surveys on the Coast Guard cutter Healy, was that they found extremely high concentrations, some of the highest ever seen of the Alexandrium cyst in the Chukchi Sea. The plot there with the red and orange concentrations. You're talking about 600-kilometer distance of high concentrations of cyst. And this is the same time when we were seeing that warming in 2019, and that continued warming up in the Bering Sea that had extended from past the Pacific heat wave years. So one of the things that is that has also been found in the past few years is the amount of saxitoxin and domoic acid, particularly saxitoxin, that's showing up around the state in marine mammals and seabirds. So that's Kathy Lefebvre's work, report in 2016 that came out, and subsequent work from then. And it's really showing lower concentrations but wide distribution, particularly up in the Arctic, which is a concern for subsistence use. So that's a big issue for us to grapple with in the network right now, as well as we're kind of waiting to see more issues with domoic acid poisoning. We have Pseudonychia blooms in the state, but not not toxic. Is that going to change? When is that going to change? Same thing with cyanobacteria and freshwater HABs species. So in addition to the coordination through the harmful algal bloom, or AHAB network, we similarly have coordination through the Alaska Ocean Acidification Network. And Darcy Dugan with AUSE is the primary organizer for this network. And I should mention that both of the networks are coordinated through the Alaska Ocean Observing System. Websites are hosted there, and they really provide key coordination and support for these collaboration and coordination efforts. The OA network is a little bit differently oriented, is that it really is about, has a focus on information exchange, connecting scientists and stakeholders, trying to share best practices, develop strategies for adaptation, improve monitoring, really get the best. It's been really fantastic to see how some of the new technology has been able to advance what we're able to measure in coastal areas in particular, and I'll show you some of those measurements. But it really has been a resource and data hub for information across the state and connecting, as well as connecting stakeholders with experts. It's been a large focus of that effort. And again, stepping back to Ketchikan Bay, I'll show you some just to reorient. So I'll show you some data that's been collected at the water quality monitoring station, as well as ocean acidification measurements that have been made by Amanda Kelly with UAF up at Bear Cove, as well as in Jackaloff, which is near Kasitsna Bay. Let me see if I can actually make this work. So Kasitsna Bay, so both in the outer bay at Kasitsna Bay as well as in the inner bay up at Bear Cove. And so this is from the water quality monitoring station at Seldovia Harbor. It's the pH data going back to 2001. The black line, the monthly averages is the black line, and then the maximum monthly value is in red, minimum value is in blue. And so the ranges are from 7.4 to 8.3, 8.2. So there's mainly showing you the large ranges. These are not, these were YSI sensors. There was a lot of concern about the accuracy of the ranges and things like that, which is true. But we've had what's been interesting to see is how well these ranges have held up in more accurate measurements. So this is an example of, and if you don't have it on full screen, Jen was reminding me this is a good one to put on full screen because of the detail here. But these are some measurements made recently, so it's a 2017 to 2018 time series by Amanda Kelly with UAF. So she has put in the CFET continuously monitoring sensors and is calibrating them 4 times a year so that to keep the high accuracy. And so we're getting really, really well resolved data. So it's 3-hour sampling. And just a couple things to point out here is those seasonal ranges in the pH units from 7.7 to 8.3. So these are some of the largest ranges of that are they're on the upper end of what you see in coastal areas. On the bottom right, the plot, I just want you to point out that that's a snapshot of time showing you tidal variability. So the up and down movement is actually the changes with the tide. So they're quite significant. So large seasonal ranges and tidal ranges in pH and carbonate chemistry conditions. And so if we then look at that in conjunction with temperature and salinity and oxygen, which are also being measured at the same time, one of the interesting things that Amanda's found and has a paper in review right now at LNO on that is really, so the pH values are here, so it's colored for pH. So lower values are blue, higher values are yellow, and then plotted against oxygen, salinity, and temperature on the bottom. And so what you'll see is if there's a relationship, then you would expect as oxygen increases that you would see a change in the pH, which you do see. And so that's an indication that photosynthesis is driving this summer pH increase as oxygen increases with photosynthesis. You don't see that same relationship actually with either salinity or temperature, which was a bit surprising, I think, to all of us, the degree to which that didn't happen. And then, so that was Bear Cove, which is up the head of Ketchmack Bay. Similarly, we can look at Jackaloff, which again was in the outer bay by our lab at Kasitsna Bay, just one bay over. And again saw a similar pattern. So again, it's pH colored with oxygen, salinity, and temperature. And again, the strongest relationship here was with oxygen, again indicating the effect of photosynthesis. And so photosynthesis was the most important, biology driving the system, but tide variations were also quite important. And then the degree of daily variation was, up to a half a pH unit. Pretty extreme. So now jumping up north, this is some similar measurements that Amanda's been making in the Beaufort Lagoon ecosystem. And here, just a sense of, these different systems and what it looks like. So this is now a time series from September of 2018 to August of last year. It's pH on the top, temperature in the middle, and salinity on the bottom. And so you'll notice that there's bring the right cursor over here. So you have the area of low pH, which is in the winter, the temperature dropping down. This is the winter ice, so indicating that you've got winter ice cover. And if you look at the salinity units over here, you're up to 37 PSU. So this is hypersaline conditions, but they're extremely productive. And so, again, you're getting biological processes happening even under the ice, which is then changing pH conditions seasonally. So the level of detail that we're able to get with some of these new measurements is really exciting. And then just stepping back to the networks and really that they've been improving statewide coordination, monitoring, and response, but we're in the early stages of that. And we really have a lot of strength in the connections that we have, particularly with stakeholders and with the management agencies, and with these really tight connections here. But we need, as well as with industry, but we need more capabilities for monitoring technology in particular and modeling. And that is bringing me to Beth's request for the grand challenges. And I will say there's way too much text on here, but I wanted to give it to Beth fairly directly for what she needs for the workshop. So I framed this in terms of the questions we get from stakeholders, right? So, the thing that if I'm talking to Margot, or I'm talking to our friends of the oyster farmers, or I'm talking to some of the state managers, either for aquatic farming or for public health, it's sort of like, hey, what's happening right now? What's the current conditions? Like, what's going on? How are these HABs or OA going to affect the species that I care about, that I'm harvesting, that I'm managing, and do I need to be worried about it now? If I don't need to be worried about it now, when do I need to be worried about it? Because is this the thing I need to be concerned about, or do I need to worry more about that next heat wave coming in, or that the other factors? So it's putting this together. So where I went on the needs is, as I showed you, we're quantifying the variability in coastal waters much better, and so we've taken a huge leap forward there. And linking that to the areas where, really to the stakeholder applications and needs. So we're doing it in certain places, but really getting that targeted to where where the uses are. And a specific need on that is, on the HAB side, is for accurate, cost-effective field test kits for PSP toxins. That is a huge need in the state, as demonstrated kind of by what's happening right now. There's a fair amount of species sensitivity research that's going on Alaska's harvested and prey species, and that more of that, and really understanding, combining that with what we know now about the environmental conditions. So both what we know about thresholds, what we know about variability from the monitoring data. And then that's, I think, that's where we're going to be able to look at synergistic effects. So really knowing the species sensitivities in relationship to those kind of realistic environmental conditions. And then going forward, really it's about modeling the environmental conditions at sufficient time and space resolution. That's a common need. Incorporating the temperature, freshwater, carbonate chemistry, ice changes in that. And then this piece of really combining the detailed monitoring, the species-specific sensitivity to create, in modeling, to create the kind of multi-stressor vulnerability assessments. We've done some in the state for both HABs and ocean acidification, but it's really putting that together to do it on a multi-stressor approach is the direction that we think we need to go. So, and with that, that's the last bit. I can go back to this, but for those who are not in Alaska, this is our 11:35 p.m. June sunset. And I appreciate the time and happy to take any questions.
Thank you very much, Kris. As we noted, please put your questions into the chat box. Looks like there's a couple of people typing already. Maya is going to read out the questions when they're ready in the chat box. While we're waiting for some questions to come in, Kris, I'm going to ask you a question since I have the chance to do that. And that was for the stakeholders that are involved in both the HAB and the OA networks. Is there much overlap between those two stakeholder groups, or are they kind of separate audiences?
They're very overlapped. So I would say there's a whole group of people that are, and it's kind of convenient in some ways. Yeah, there's a lot of overlap, especially between state agency managers and researchers that are tapped into both. There are the public health agencies tend to be more mostly on the HAB side right now, and that reflects the timing and the immediacy of the threat. So HABs we are dealing with right now and have been and will be. Ocean acidification, we have the conditions out there. You know, we're close. It's not as an immediate of a threat, and it's a different question. It's more a resource management question than public health question. So that would be the big difference. Otherwise, other than that, there tends to be a lot of overlap.
Thanks. I'm going to let Maya read the questions, and please, all of you, go ahead and type your questions in. If you want any follow-up, you can raise your hand, and Jen will unmute.
Thank you, Beth. I saw a few participants typing, so I'll just wait until we get our first question up in the chat box.
Looks like there might be one from Astrid.
So I see one on cyanobacterial. Yeah, so I'm not so we have concerns about cyanobacterial issues for the freshwater lakes on the North Slope in particular, so out of Kotzebue and Uktiavik, so in the North Slope communities. I'm I don't know the current status. We've had one of the things that's been great is the folks from our in-cost Charleston lab have done recent training on freshwater freshwater HAB identification. So it's been helpful capacity building within the state. And I see the note from Joseph Ortiz about remote sensing products. The remote sensing products we're using right now are in the Gulf of Alaska are mainly for temperature, because the blooms, the Alexandrian blooms it can't be detected with ocean color and things like that. But we are using, because of the tight relationship with temperature, and the thresholds that we've been able to establish over time, both from the work in Southeast and South Central. With that, we've got a sense for when conditions would be favorable. It doesn't mean that you will have a bloom, because there's other factors at play. So we can use satellite sea surface temperatures for that. The ocean color possibly could be for cyanobacteria blooms up north. The concern there is that you have relatively small lakes, and you may not have the spatial resolution. They may be too small for some of the satellite products.
Thank you. Our next question is from Joseph Ortiz. He asks, when working with DO, temp, and pH sensors, do you correct the DO for temperature changes to tease apart the temporal dependence of O2 from the biological part?
Yes. So that's one of the things that it's really great to be working with the chemical and ecological oceanographer like Amanda. That's one of the things she's paying very close attention to. And I was hoping that she would actually be able to join this call, but she's actually doing field work over at our lab right now. Yeah, and I would love to talk with you about remote sensing imagery. That's the world I came from back in DC. So always interested in that.
Thank you, Kris. We have some more typing from our participants, so we'll wait for our next question.
We do have a comment from Shelley. She says the Cyan project is delivering cyanobacteria products from OLCI at 300 meters.
That's right, and we are getting the products up in Alaska. Yeah, hey Shelley, do you want to tap in? Do you know how many of the lakes that I want to say I heard that in a discussion that are large enough for those products, but I can't remember the number.
Shelley, if you want to chime in, you now have a microphone option at the top of your screen. And if you click on that, you can speak if you'd rather do that.
Okay. Hey, Kris, can you hear me? Hello?
Yes.
Okay. Yeah, Kris, I do not know off the top of my head for Alaska. You know, obviously clouds are an issue, but as far as resolution, I can find out how many lakes. I know across the whole U.S. there's about 2,000 lakes total that we can resolve.
Something like that, yeah, because I saw it, I want to say it was one of the discussions I saw it, and I just can't remember what it is. I probably have a PowerPoint sitting somewhere that has it.
Okay, yeah, I can definitely get that number. Sachi would have it. The other thing, too, is we are starting to develop some Sentinel-2 products, which gets you down to 20 meters. They're, you know, not completely online, but we're at least playing around with them for the Chesapeake, putting them out there. So that's an option coming down the line.
That's exciting.
Yeah, more of like a bloom index, not so much a cyanobacteria specific thing, but you know, an abundance of chlorophyll kind of a thing. So anyway.
I see a discussion at the August workshop around this.
There you go.
Looks like Jesse Turner is typing something, so we'll see what that is when it comes up in the chat box.
Yeah, Jesse. So Jesse mentions that specific momentum for a statewide changing conditions vulnerability assessment. That's what we're talking about. In fact, Darcy and I were just talking about this earlier, a couple of days ago, as a direction that we want to go, because we're in, if you think about when we did the first kind of vulnerability assessments, where it was based on the information we had at the time, and we had a lot less information in particular about coastal variability. And so now we're in, both because of what's being done for species, what has been done for species sensitivity, as well as that better understanding of variability on from daily to seasonal to interannual timescales. We've got, I think, the opportunity to do that. Anyway, it's I think it's something that the networks are going to take on. We have to figure out how and who and when.
If there are any other questions?
We are always looking for additional capability up here, because while we have a huge state, we don't actually have that many people. And so if folks are interested in working up here, we always love the help. So that's a plug for hoping that the folks will get interested in Alaska, these issues up in Alaska, and come help us out.
Yeah, not to mention all of the nice pictures that you included in your presentation, Kris. That's definitely attractive.
There's lots of reasons to be attracted to work up here. No, I mean, it's pretty fascinating. I've appreciated in the other webinar talks is kind of looking at where things are there, and I was sharing this with Beth earlier, that it's like, dang, I want all those tools, especially on the modeling side. But the exciting point is we have that to do, and we're getting into the position where we can do some of that in ways that we couldn't before.
All right. If there are any other questions, go ahead and enter them into the chat box. In the meantime, I wanted to let you know that we are going to be posting a recording and a PDF of the slides on our workshop webpage. And Jen will put in that address into the chat box. That's also where you're going to find the Google form. You can provide other input and comments on that Google form. We really would value that, because it's going to help guide our discussions at the workshop. We are concluding our webinar series. So if you're on the list for the virtual workshop in August, look for an email soon with a registration link for that. We're going to be asking you what kind of breakout sessions you want to be involved in, and some other questions about your level of involvement. We're also going to be posting an agenda on that workshop website soon. Thank you very much for joining us, and we hope that you enjoyed the whole webinar series. We certainly learned a lot from hosting it, and we look forward to discussions at the workshop.